Hello, everyone, and welcome to this webinar hosted by Jen. Our topic today is Advances in Cell and Gene Therapy, Characterizing Large Viral Vectors Using Macromass Photometry. Today's webinar is sponsored by Refeyn. Thanks so much for being here. I'm Uduak Thomas, Senior Editor with Jen, and I'm delighted to be your host for today's webinar. Our speaker today is Dr. Laura Pala, Product Manager at Refeyn. But before we get to her presentation, let's go over some background on our topic for the day. Accurate characterization of large viral vectors is a critical step in developing viral vector-based gene therapies and vaccines for use in cancer immunotherapies, genetic disorders, infectious diseases, and more. This process is indispensable for ensuring the safety, stability, and efficacy of biopharmaceutical products. However, characterization methods are often time-consuming, requiring multiple orthogonal techniques. In this webinar, Dr. Pala will discuss how Macromass Photometry enables fast qualitative analysis of adenovirus and lentivirus samples for cell and gene therapies. She will introduce KaritroMP, a novel analytical instrument that uses macromass photometry to characterize large viral vectors in research and development environments. She will also share case studies and benchmarking data using adenovirus and lentivirus samples during the webinar. Key learning objectives for today's webinar include the advantages of using the KaritroMP system to characterize viral vectors at a single particle level based on contrast and particle size, how the KaritroMP system can identify empty or fragmented capsids in adenovirus preparations and assess the purity and quality of viral vectors, how macromass photometry can be used to compare different production and purification methods, as well as assess variations between batches. And lastly, you'll learn how to integrate the KaritroMP system throughout the cell and gene therapy development process. All right, I'm going to hand things off to Laura now.
Thank you very much for the introduction. I am really excited to be here today to announce the market launch of our new instrument coming to market for characterizing large viral vectors using macromass photometry. This is called the KaritroMP, and it is a new benchtop instrument that allows for rapid characterization of large viral vectors and can specifically assess lentivirus and adenovirus purity and stability within minutes. We are really excited to announce that it will come to market really soon, and specifically in Q1 2024. This is based on a new technology, which is called macromass photometry. And this technology is an evolution of Refeyn's core technology, mass photometry, which was, let's say, tuned and adapted in order to be able to measure particles in the nanometer range, which includes large viral vectors such as adeno and lentiviruses. I would like to tell you a bit about the technology and how it works. So this is basically an optical measurement that happens at the glass-liquid interface. We have a glass slide on top of which we deposit a droplet of liquid containing the viral vector samples. And what we do is sending a light source from the bottom. So once the sample is illuminated, part of this light is scattered back by single particles, and part of this light is reflected by the glass surface. So what we measure is the interference between the light scattered by each and every particle and the light reflected from the glass. This signal is recorded in form of contrast, and because it's basically related to the scattering of the particles, is also related to particles' mass and refractive index. So when we have particles of different masses, they will be measured at different contrasts. So smaller particles, or let's say particle with lower mass, would have a lower contrast signal compared to particles with a larger mass. So this is one of the two parameters that macromass photometry measures, the contrast or proxy for mass. The other parameter is the particle size. This is obtained by sweeping through focus, which means moving up and down the glass slide in order to measure the vertical dimension of the particle size on the glass. Now, if we are working with spherical particles, this vertical dimension would correspond to the particle diameter. Of course, if the particle is non-spherical, this would be only one of the 3 dimensions, and specifically the vertical one, depending on how the particle lands onto the surface. This can be calibrated for size, and effectively the output would be nanometer diameters for single particles. So at the end of the day, what we have is the measurement of 2 parameters simultaneously. The particle contrast, or proxy for mass, which is obtained by measuring the interference between the light scattered and reflected, and the particle size, which is obtained by recording throughout a vertical sweep on the glass slide. So we measure these 2 parameters, and how do we show them? How do we represent them? You can see an example of graph produced with the KaritroMP here on the bottom left. So we have a 2D plot showing you on the x-axis, so the horizontal one, the contrast distribution for single particles. So on the top of it, you can see a histogram showing you the contrast values for single particles. On the vertical axis, so the y-axis, you can see the size information. So here again, you have another histogram showing you the size distribution for single particles. In the center here, you have a 2D plot, which is represented in form of scatter plot with kernel density. This means that every point, every dot would correspond to a particle measurement value with specifically one contrast and one size value. And in region in the plots where you see darker areas, this means that you have a higher concentration of data points. In this specific graph, what we have is a measurement of full empty and fragmented adenovirus capsids. So what we have, if you look at the vertical axis here, is one main population at about 80 to 85 nanometers, which correspond to the adenovirus capsid, and some smaller items, some smaller particles present in there, which would correspond to the other fragments or also protein aggregates. So if you look at the main peak in here, so the adenovirus capsid peak, you can see that underneath that, you have 2 main populations in terms of contrast, basically particles that have different mass. And this would be effectively the full capsids and the empty capsids that cannot be distinguished based on the size measurement alone. So effectively, with the KaritroMP, what you can do is a fast, simple, and qualitative analysis of adenoviruses and lentiviruses, because you can distinguish multiple populations even if they have similar or overlapping size. You can also look for relative changes between samples in process, so we are able to distinguish from crude to purified samples. And you can use this information to quickly assess the purity and stability of the samples before taking a decision on the next step very quickly. This technique is quite good to look for differences and similarities, and therefore compare different data sets. So you can use this to optimize production, purification methods, and also to assess batch-to-batch variability. I would like to highlight here how the contrast information can be used to resolve particles that have comparable size. So what we have in this graph is our size calibrant measurement. This is called sizeference. This is an equimolar mixture of silica beads that have different size, different diameters, ranging from 50 nanometers to 120 nanometers. So as you can see in here, if you look at the vertical axis, so the size information, this histogram shows a broad population, but really not a great resolution, if not at all, of the size populations present in here. However, if you look at the contrast axis, you can see 5 really nicely distinguishable peaks in there, and you can even resolve the 50 nanometers beads from the 60 nanometer beads, which is quite powerful. So what you can effectively do with the Karitro is take advantage of the mass differences that would be reflected in the contrast axis in here in order to distinguish populations that, based on the size alone, you wouldn't be able to resolve. Now, if we look at the comparison of the size measurement obtained with the KaritroMP with orthogonal techniques, this is what we would get. In here, what we have is a comparison with of the Karitro data with nanoparticle tracking analysis, or NTA, and dynamic light scattering, or DLS. So first of all, let's focus on the Karitro data. So in here, we have two histograms in gray and in blue. So the gray histograms would correspond to the size measurement obtained with the KaritroMP of the 5 different populations coexisting in the size-Ferenc calibrant. In the blue, in the shades of blue, what you can see is the resolution of the 5 populations based on their contrast values. So basically, we are identifying where the single population would appear based on their resolution in terms of contrast. What you have in here is also an orange line showing you the NTA data, and the green one showing you the DLS data distribution. So there are some differences in between the various techniques. The main, so the first difference that you may observe is the fact that the data with NTA and DLS appear larger compared to what we measure with the KaritroMP. This is due to the fact that both NTA and DLS measure the hydrodynamic diameter, which takes into account the diameter of the particle, but also the hydration shell around this particle, which means the layer basically of solvent that is around it. While the KaritroMP measures the geometrical radius, which basically is only the diameter in this case of the particle itself, without taking into account the hydration shell around it. So in that case, in that sense, both NTA and DLS would give larger size values compared to KaritroMP. Another difference in here is in terms of resolution and bias towards larger data, larger particles. So both NTA and DLS have a bias towards larger particles, which means that if small and large particles are present together, both NTA and DLS would have an issue seeing the smaller particles as well as the big ones, because the big particles would mask, let's say, the presence of the smaller ones, and effectively you would have saturation of the signal. And with the KaritroMP, you don't really have this problem, because it doesn't really matter whether you have coexisting presence of small and large particles, because there is no problem in effectively measure them and quantifying them. So what you have in here is the fact that the KaritroMP has a better resolution based on the size measurement alone compared to NTA and DLS. I would like to focus now on the KaritroMP sample carrier, which is needed for every Karitro measurement. This is composed by 3 items. So a slide, which is made of glass coated in order to allow for an optimized landing and the immobilization of viral particles specifically. Then we have a 14-well cassette, and we have a plastic lid. The reason why we designed the system in this way is to allow for a closed system. So effectively, what it means is that the operator would be preloading the cassette, so the sample carrier, in a safe environment, such as biosafety hood. And then once this is loaded, it can close it with a lid and transport it outside of the hood and on top of the instrument. Now, the reason why we design it in this way is to prevent any cross-contamination or any additional risk for the operator, taking into account that usually these viral vector particles are BSL2 or CSL2, so present some sort of biohazard for the user. So in this way, we are removing or reducing, let's say, the exposure risk. Another advantage of this consumable is the fact that it allows for a semi-automated workflow. So again, there are 14 wells in here that would need to be preloaded by the user before placing the sample carrier onto the machine. And these wells need to be loaded in this way. So we need one well used for the calibrant measurement that allows for size calibration. And then you can use up to the other 13 wells for measurements of samples. Once this has been preloaded, the cassette has been basically closed with the lid and transported on top of the machine. Effectively, from that point onwards, the user is not required to have any input into the measurement, so allowing for a semi-automated workflow. The well dimension allows for a very little sample consumption. We are talking about 5 microliters of sample at the optimal concentration of 10 to the 8, 10 to the 9 particles per milliliter. And this consumable, this sample carrier would help obtaining a measurement of a full cassette in a fast manner, because it would take up to 1.5 hours for a full cassette measurement. Now, if we look a little bit more in depth into the workflow, we have again a 20 minutes hands-on maximum, and following by 1.5 hours automated measurement. Looking more in detail what the hands-on operations are effectively, we have an initial stage of sample prep, where sample need to be diluted to the approximate 10 to the 8, 10 to the 9 particles per milliliter optimal concentration. Then the user would need to manually load the calibrant and the sample into the wells. So this is requiring only manual pipetting operation. Then the sample carrier needs to be closed with the lid and transported outside of the biosafety hood and placed on top of the instrument. At this point, the user needs to set up the instrument and the software, basically, so the measurement protocol, as well as basically define which sample is contained in which well. At that point, the software would go by itself, basically moving from well to well and measuring the 13 sample plus calibrant. The full time for cassette measurement is, as above said, 1.5 hours. And while the data are acquired, the results can be shown on the fly. And at the end of the run, they can be exported either for post-processing with the instrument software, or they can be also exported in a CSV file if needed to be post-processed with alternative software or programs. I would like now to focus on data on adenoviruses and lentivirus samples. So what we are showing in here is an example of adenovirus purity measurement. We have a mixture here of the chromatographic fraction coming from purification methods, where we have a mixture of the full capsids, represented by the gate B, and the empty and fragmented capsids. The empty capsids are enclosed in the gate A. So what we have in here is, again, as mentioned in the introduction, one main population corresponding to the expected size for adenovirus particles, so around 80 nanometers. And underneath that, you can see 2 main populations with different contrast, with different mass. So the B8 is the full one, which is present in 52% basically in the total adenovirus region. And then we have the empty population, which is about 48% of the total adenovirus region. Here you can also see a plus minus 1% for both of them. This is the standard deviation of the counts within the region over triplicate measurements. So this is telling you that the relative counts between empty and full is quite reliable if you perform the measurements multiple times. The conclusion of this is that you can use the KaritroMP during R&D and process development to effectively optimize the adenovirus production. We are talking about early stages, preclinical phase. We are not talking about manufacturing here, but effectively this tool is quite powerful in order to have within minutes an answer about how the production is going, and even compare different production methods. I would like now to focus on the adenovirus stability measurement here, and what we are doing is a thermal degradation of adenovirus sample. So we've been heating the sample at 47 degrees Celsius for about an hour and monitoring what is happening to the adenovirus sample itself. So at the 0 time point, what you see is about 47% of the counts in the gate A, so the the adenovirus region. And one main population present there that would tell you that the sample is quite pure. After 15 minutes of thermal degradation, you can see that the number of counts decreases to around 18%, and new populations appear at different size and contrast, telling you that something is going on with this sample and is effectively potentially degrading. After an hour, you see that the number of counts of adenovirus particles in the gate A is potentially reduced to zero, like it's around 0.3%. So it's almost, we have almost no particles in this region. And we have an increase in the particles in the other regions that appear during the degradation experiment. So what we can conclude from it is the fact that the KaritroMP within minutes can give you a quick assessment of the presence of populations during to degradation and an assessment of the stability of the adenovirus sample. And once this is done, the operator can take a quick decision on how to move on and what to do in the next step, what to do with these samples. Now I would like to move on to lentivirus samples. Lentiviruses tend to be more complicated compared to the adenovirus samples that we talk about until now. Reason is the fact that they tend to be much more heterogeneous, and the isolations of distinguished peaks is more complicated compared to the adenovirus samples. If you look at the blue graph in here on the left, what you can see, for example, is a main population that is at low size and low contrast, and then another population that can be seen as a resolvable peak at above 0.2 contrast. So we had reasons to believe, after multiple experiments and research, that the lentivirus population is actually coming here in this region, which is not the majority actually of the data points measured in here. These correspond to about 11% of the number of counts. And the other data points that are present at low size and low mass are effectively extracellular vesicles, as well as the subviral particles and also large protein aggregates. Of course, we needed a way to prove that. We needed a way to find prove that this was exactly the case, and also to be sure that the lentivirus population are represented by the gate A, so that we can apply it also to all other lentivirus samples. So the way we approach this with our collaborator is basically by measuring, in addition to the lentivirus sample, 2 negative control samples. In the first one, we have a mock transfection sample where basically we are transfecting with a plasmid that does not code for the lentis. And what you see is still the presence of the population at low size and low contrast, but virtually no particles in the gate A. In the other negative control sample, what we've done is a transfection with a plasmid that does not code for the packing protein that would lead to the lentivirus formation, but still codes for the genome and the glycoprotein. And again, what you see is very similar to the other negative control, with virtually no particles in the gate A, but still the population here at low size and low contrast. So the conclusion of this is that, first of all, we are able, even though lentiviruses are quite heterogeneous, to distinguish what is lentiviruses from what is not lentiviruses, and are most likely extracellular vesicles, subviral particles, and aggregates. And also, now with these negative control experiments, we also have a proof and a reason to believe that the gate A is a good way to actually represent where this lentivirus particle can be enclosed and also relatively quantified. So by applying the same gates just defined on other samples, we could look at the lentivirus purity. What we have in here is a comparison between a crude sample and a purified sample from the same prep. And what we can see by applying the same gates as defined in the slide before, the number of count goes from 2.4% in the crude to about 24% in the purified sample, increasing by tenfold. So this is quite promising, because effectively what we are seeing is an enrichment of lentiviruses in as you go along with the purification. If we look at the benchmarking data that we got from our collaborator, these are infectious titer data, we can see an increase by about 7-fold from the crude to the purified samples. So this is telling us that the purified samples contain 7-fold higher number in infectious particle compared to the crude. So even though we do not claim that we can measure infectivity, we can still see a very nice correspondence in between the number of counts and the count increase between a crude and a purified sample and the infectivity of these samples in there. So effectively, just to clarify that, we are able to distinguish and count a number of particles that have the correct size and contrast, so correct size and mass effectively, to be well-formed lentiviruses. And these can be often informative related to the infectivity, as long as the particle with the right mass and size are also the infectious one, which in this case seems to be quite representative. So the reason why this measurement is quite powerful is because within minutes you can have a quick assessment of the number of count in the lenti region without waiting for a long time to produce infectivity data, and then move on and then decide what to do next effectively. So this is quite useful if you're doing process monitoring or process optimization as well. And what I would like to show you in this last slide is the reproducibility of the data that you obtain with the KaritroMP. What we have in these two graphs is adenovirus data and lentivirus data of the same sample that were conducted in triplicate. So we have 3 repeats for both. And what we are showing in here is a different way to visualize this data. This is a contour plot type of visualization. The reason why we decided to use the contour plot in here is because it's much easier to basically look for similarities and differences when overlaying data, in this case is repeated measurements. In order to be able to read it properly, I would like to focus on the lentivirus reproducibility data so that I can explain how to look at this type of visualization. So in the thickest line in here, you have 10% of your data points basically enclosed by these lines. And like enclosed by the, let's say, medium line, you have 40% of your data. And in the lightest line, you can have about 70% of your data inside of the contour. You have also some scatter points outside of this region, which are the points that are basically more scarcely represented in the plot. So the way to look at it and the way to look for differences and similarities is basically by assessing how much the contour line overlap with each other. So in this case, if we look at the lenti graph, you can see that 10% of the data is always enclosed by this thickest line within the same regions. And same is for 40% and 70% of the data. This shows you effectively that these data are quite reproducible, and repeated measurements show the same values in the various regions of the plot, which is very powerful. And the same can be said also for the adenovirus samples, where you have like the same values produced over the triplicate measurements. So I would now like to thank you all for the attention. I hope this was clear. I am happy to take any questions. Thank you very much.